An herbarium is basically a collection, a museum-grade collection of formerly living plant material. And I like to say formerly living because it's all dead, but sometimes it also includes fossil specimens, and so not just simply dried material, but the fossil material. At the MSU Herbarium, we mostly have dried material. We don't really have a fossil collection. But a typical plant specimen basically looks like this. So it's a dried, pressed plant preserved on archival paper. And so in addition to the plant, you also have a label that provides the fundamental data associated with the specimen. So at its most fundamental level, an herbarium is really just a museum that documents where plants grow and when they grew there. That's the basic bit of data that we have in herbarium. And of course, what you can do with that little bit of data is really pretty amazing. The MSU Herbarium has over 520,000 specimens. Most of those are flowering plants. That's where the bulk of our collection lies. That's also, of course, the bulk of plant diversity is flowering plants. Most plants that are alive today are flowering plants, so that's our focus. We have about 145,000 lichens, then various other collections of things like mosses and liverworts, what we call non-lichenized fungi, which means mushrooms and cup fungi. We have those in our collection, and a lot of other things like algae and things like that. But the bulk of our collection is flowering plants. The primary mission of the Michigan State University Herbarium is documenting biodiversity. That's the idea, is we're here to document plant biodiversity for posterity. And our geographic focus is Michigan and the Great Lakes. It's part of our job to do that. And of course, that's also where most of the researchers at Michigan State University are focused on, is the Great Lakes region. So it's also a region of greatest utility. But we have plants from all over the world. And the primary reason we have plants from all over the world is that Michigan State University is a huge place. There are plant researchers who work all over the world, and we accept their specimens for their vouchers for their scientific studies. If they're doing work somewhere and they need to leave specimens for posterity, then it's our job to take those on and to house them. By doing so, researchers can actually make sure that their science is verifiable, repeatable. Part of the fundamental scientific enterprise is doing something that other people could do if they wanted to. That's what makes science is the ability to redo it. And specimens add to that. So people can go back and confirm identifications. They could re-extract DNA from an herbarium specimen if somebody's done that. There's a number of things researchers could do, but probably more important than checking on what a researcher has done is this notion they can build on a researcher's science by coming back to the same specimens, the same material, and doing something new with it, adding on to the research and building kind of science that way. When a collector collects a specimen, it's a very low-tech, old-fashioned way. We still use plant presses. They're just sheets of lattice wood and blotter paper, which dries the specimen between sheets of cardboard to provide a firm backing for it. You put the fresh plant inside the newspaper and try and arrange it as best you can so that it looks kind of nice, because there's a little bit of aesthetic nature to a specimen if it's done right. And then you press the plant, you put straps on it, and you pull it as tight as you can, and then you try to find some place dry to keep this, preferably warm and dry, so that it dries out nicely. Of course, we have a dryer upstairs to do that. When it comes to us, it's still in the same newspaper that the collector put it in. And you can see this is a nice new specimen, beautiful and green. And this is what they look like. They're a little delicate, but they should be paper thin, pressed flat, and ready to be attached to a piece of mounting paper. So our mounting paper is archival quality, pH neutral, and we simply use a pH neutral glue, also archival quality, but it's basically like Elmer's glue, to glue the specimen onto a piece of mounting paper, glue the associated label, try to arrange it in a nice way so that it's both kind of an attractive specimen, but most importantly, is the highest quality for science. And so the idea is that ideally you'll have some leaves turned up and some leaves turned down, some flowers facing up, some flowers facing down, so that you can see the back of all the kinds of plant parts. Once they're glued, the glue can kind of seep out and stick to things, so we just use a simple piece of wax paper to cover it. And then on top of the wax paper, we put weights to make sure that while the glue is drying, the plant stays attached to the paper. And then we just stack the specimens up one by one until we are done for the day. It's that simple. Once they're actually dried, we just disassemble that, and then they're ready to be accessioned into the herbarium. There were a couple of early directors in the first few years of establishment of the herbarium. But then in the early 1870s is when the university hired Dr. Beal. And William Beal actually was the first herbarium director to really focus on growth of the herbarium. During his time at Michigan State University, the herbarium grew from about 20,000 specimens to over 100,000 specimens, so over 5 times in just the 40 years or so that Dr. Beal was a faculty member here at MSU. So that the herbarium at that time actually became one of the largest herbaria in North America, a very valuable herbarium. Dr. Beal had trained at Harvard with Asa Gray, who was called the father of American botany. And he made friends with botanists all over the country. So at that time, we were getting a lot of historical specimens from the West Coast and the East Coast and all parts of the United States from Dr. Beal's friends that he made during graduate school. But he also convinced the Board of Trustees to actually buy collections. It's a practice we don't do any longer because there's some ethical problems with buying and selling, plants, especially things like rare plants and things. But nonetheless, he convinced the Board of Trustees to buy a lot of specimens. So we also have a lot of important collections that people had offered for sale back around, say, the turn of the century. Herbaria and plant specimens in herbaria are the foundation for what we know about plant species. When you describe a new plant species, it's actually a rule that you have to cite an herbarium specimen of that species. We call that the type specimen, the sort of original type specimen. And that's how we describe species, is by taking a specimen of a plant and comparing it to other known and identified species or samples of that plant and seeing if they're different. There's a lot of science and now statistics and sometimes even genetics involved in it, but basically it's comparing one specimen to another and to describe new species. And then the other things herbaria focus on are floras. For a particular geographic area, what plants grow there. That's important if you want to know what plants grow in Michigan, for instance. But one of the new things people are doing lately is studying more about how plant distributions change. It's really important with climate change, with invasive species, with habitat degradation, to know what used to grow here and what grows here now to see how we're actually changing our planet. So for instance, one of the projects the herbarium has had over the last probably decade is a study of plants grow at Baker Woodlot. So you may know Baker Woodlot, 70 acres just to the south of us in the herbarium. It's a beautiful bit of native forest in Michigan. It's never been clear-cut. It was selectively harvested, but never clear-cut. And we just finished a study, built on a study 40 years ago, that shows how the plants have changed at Baker Woodlot. So we've documented that some plants have gone locally extinct, what we call extirpated. They used to be there 40 years ago, but they're not there now. Those are things that even were common, like for instance, there was the woodland phlox there 40 years ago. It was listed as common then, it doesn't grow there now. But also a lot of rare species. The lady slipper orchid that was there is no longer there. And then we also have documented a lot of invasive species moving in. 40 years ago, garlic mustard was not known from Baker Woodlot. And now, of course, you can't walk out there without stepping on it. So those are kind of the traditional uses of herbaria, is what we call floristics and taxonomy, documenting what grows where and documenting new species and extinctions too. tell a little story, may be relevant. This is my supplemental research space. We're doing a big project upstairs, and I got kicked out down here. But I wanted to talk a little bit about kind of the multitude of ways that we use specimens. I'm going to put these over here. In ways that sort of aren't traditional uses of specimens, because that's one thing that I think is important. Yeah. So these specimens are from my own research, and one of the things I study is plant biodiversity. I'm really interested in new species, where they grow, and how they differ from other species around them. And this particular species we're looking at is actually an undescribed species of plant from Bolivia. And I first became aware of it because I was writing a treatment for the flora of Bolivia for this plant group that I love. I focus on Central and South American plants largely for my research. And the editor of this particular treatment that I was writing, so it was in the last stages of the manuscript, and he sent me a note by email that said, you know, I saw that you included this species, but I think you need to include that one. Here's a picture of a specimen. I didn't see that you had seen that. And so he sent this picture, and it was obviously a new species that I had never seen before. And so he and I are now collaborating on describing this species. One of the reasons why I wanted to show you this new species that we're describing is because one of the things I do is look at species in their evolutionary context. And to do that, you need to build an evolutionary tree. And we build evolutionary trees now using DNA from plants. And it turns out that you can actually extract DNA from herbarium specimens, sequence it, and then use that to build these evolutionary trees. We call them phylogenies. And so this is a specimen that this researcher in South America, his name is Stefan Beck, sent to the United States National Herbarium, which is part of the Smithsonian, a few years ago. And it happens to be the one that he sent to me by email and said, have you seen this? So when he sent me the email, I borrowed the specimen from the United States National Herbarium and asked them for permission, took a little bit of leaf material. You only need just like a paper hole size punch. Took to the lab, extracted the DNA from this very specimen. So because we could get the DNA from this specimen, I can include it in this evolutionary tree I built of this particular plant group. So it's just one of many things we can do with specimens that we couldn't do when specimens were first collected. And that's one of actually another part of my research is scholarly use of herbarium specimens. And the idea behind what I do is I track the changes in how specimens are being used in research over time. So for instance, with some other collaborators, I published a paper last year where we did a literature review of over 13,000 papers and used that with time as a variate. So we looked at these 13,000 papers that were published over 100 years that used the word herbarium inside the paper and used artificial. So we looked at these 13,000 papers over 100 years using natural language processing and some artificial intelligence that allows us to artificially categorize these into categories that don't reflect our biases. These are based on the words in the papers. The computer can actually segregate these into categories, and then we can name those categories, and we can show that, for instance, 100 years ago, most papers using herbarium specimens were describing new species, and they were talking about what plants grow where. But now those are just one of a multitude of uses, including things like using specimens to build phylogenies, also using specimens to document changes in floras over time related to climate change, using specimens to build an environmental niche for a particular species, so you can see what environmental variables correlate with where species grow to understand how climate affects where they grow. There's a million things people use herbarium specimens for. It's kind of an exciting thing to see those changes over time. So these are just a few of our seeds, and I mentioned earlier that I study plant diversity. One of the reasons why I like to study plant diversity is because it's just the most fascinating thing on the planet, right? So when it comes to seeds, there are a lot of things that control the different kinds of adaptations the seed may have. But two of the major ones really are dispersal, how the seed is going to get from one place to the next, and then also early life history, what happens just after a seed germinates. So for instance, if you're a forest tree and you are going to have to grow in an area that is deeply shaded because all of your parents are growing above your head, or an area that is really dry, you may have to have a lot of resources in your seed. And that might mean you have to have a bigger seed because you have to have more nutrients. So things like seed size are partly controlled by what happens in those early stages of growth. How much resources does a particular plant need early in its lifetime? But I think dispersal is really where a lot of the cool stuff happens. So dispersal in plants can happen in any number of ways. And plants have been really pretty clever about coming up with ways to be dispersed. So for instance, think about a dandelion. Now technically what you're looking at when you think about a dandelion is a whole fruit, because the seed is just something inside of the tiny black part. But each seed basically has its own parachute, and all it takes is a little bit of wind and those seeds can blow forever, right? They can really travel a long ways. So that's a really simple way to get around, but it's actually a really complicated apparatus to do that, to actually have the appropriate development to build that little parachute is pretty complicated, pretty fascinating. There are a lot of other plants that have those kind of plumes. So if you think about a milkweed seed that has all the giant hairs, different plant families, different plants have these adaptations for blowing their seeds around, and they can be different ways. Some plants just have wings, so they may not have a plume, but they might be flattened and they might have a thin membrane around them, think like an ash or a maple. Again, those are technically fruits, and inside there is the seed, and they've grown for each seed has its own fruit, it's shaped like a big wing usually. So that's another way of dispersing. But there's a million other ways. Think about like a cocklebur or a sandbur that has all the spines that stick in your socks and get dispersed by animals. Stick tights are another great example of that. And the little bed straws that bloom in the spring and get the little round things that get stuck on your dog or your socks, also annoying. Basically, that's where the idea of Velcro came from, is they have these little curled hairs on the end of them, and they get into your pants and they want to stay there forever. And so there's those ways. There's also seeds that just develop a sticky exterior. So the seeds themselves actually just stick to things and move around that way. There are all sorts of seeds that are digested to go through some kind of animal's digestive tract. And then the advantage of doing that is, firstly, you just get eaten and your seed goes wherever the animal goes. But also, of course, as soon as the animal excretes the seed, each seed comes with its own pile of fertilizer, right? So think about a pile of cow dung with any kinds of seeds in it. So if they can manage to pass all the way through a digestive tract and still be fertile, then they're in great shape for the next generation. There are seeds that are dispersed by water that float. The best example of that is probably a coconut, right? So coconut trees grow right on the beach. They hang out over the water. They have these gigantic fruits, but again, there's one seed inside of each fruit. Gigantic fruits that fall into the water. They just wash around to the next island and grow there, and then the coconut palms have dispersed to another island. One of the major foci of the herbarium these days is digitizing our specimens. By digitizing, what we mean is putting an image of the specimen online and then capturing all of the data from the label and parsing that into fields online so people can search for plants by the genus and species of the plant. They can search by geography, they can search by date, they could do some combination of things, they can search by collector. Anything that's on the label, they ought to be able to search for. So that's, of course, a huge push right now in herbaria and natural history museums, probably all kinds of museums across the globe, both the EU and the US National Science Foundation, as well as a lot of other countries too, have put a lot of money into putting these biological data online. There's a global biodiversity information facility that is run by the EU out of Denmark, and their job is to consolidate all of these data, so it's kind of one-stop shopping. Anybody can go there, search for collections from any repository that is putting their data online, and find what they need to study. So here at MSU, we are capturing our images and recording those data into our own database, and then that feeds into these portals, these online portals that have access to the data. So the reason we do that is partly because there are over 600 herbaria in the world, and you never know for sure where plants are going to end up or who's going to have plants that you're interested in for your own research or your own educational needs. And so if everybody contributes and it's one-stop shopping, experts from around the world can search for plants. So for instance, this plant in here now is a collection of the genus Monarda bee balms from Mexico. So you wouldn't necessarily think, if you were studying Mexican Monarda, to look in the herbarium at Michigan State University, but this specimen actually happens to be a type specimen, the original specimen used to describe the species. So it's a really important specimen that could go overlooked. So by taking an image of it and putting it online, it allows anybody to have access to that. It's opened up a whole world of research possibilities by having all these data online. So for instance, people are really interested in phenology, the timing of reproductive events of plants, and how that's changing with global warming. So, for instance, are plants flowering earlier in places where it's getting warmer? We find that to be true, but it's only by having these huge repositories of data that we're actually able to test that, where we get enough specimens from enough places to be able to know that, in fact, that's actually happening. So I just looked before you guys came for this interview, and so far this year, there are 87 citations for 2020. The year's not up yet, and not everything is cataloged anyway. But for the specimens we have online, for the year 2020, there are 87 citations of our digital data. Published scientific papers already in 2020 that you're using our data for different scientific studies. So 87 papers, of course, is an enormous amount of research that we've enabled by putting these specimens online. These are sort of educational amateur booklets that were published in the late 19th century. And the idea was students, mostly students, probably junior high and high school, could make their own plant collections, and this little book provided everything you would need for that. So instructions on the front, a few pages about how to identify plants, and then once you get into the meat of it, there's a page where you can fill out information about the plant, where you found it, what it looked like, and describe it. Each little separate booklet has one page where you can write information about the plant, where you found it, when you collected it, the description of the plant. And then inside the little sleeve, you can actually put a little image of the plant. So there's actually the specimen of this little, looks like a spring beauty. And this was collected Kalamazoo, Michigan in 1894, so roughly 125 years old from Michigan. And so what this does, it actually can help us understand what was growing in Kalamazoo, Michigan at this time. These are things that weren't originally deposited in herbaria, but that actually people donated to us when they found them, say, in their grandmother's attic or that kind of thing. Dr. Beal had a really lasting legacy at MSU. From my perspective, I think the thing that was most special about Dr. Beal is that he was really invested in the future. He did a lot of great science on the short term, but his real legacy is actually focused on long-term biology, long-term science, long-term infrastructure of science. So when I say that, what I mean is, he was the first really important director of the herbarium. He grew the herbarium over 5 times at a time when the herbarium started off pretty good size already. So he had huge impacts on the herbarium, and now the herbarium is over 150 years old. He founded the Botanic Garden, another really important research resource on campus, also almost 150 years old. He started his long-term seed viability experiment, and that's the longest scientific experiment. So he really had his eyes to the future. And, an important plant science institution like Michigan State University, with so many plant scientists doing so many different kinds of plant research, relies on these resources that Dr. Beal established and built. And so I think that his legacy is just these contributions to the infrastructure of science that are ongoing. You can look at him planting the pine needle, for instance, still, in existence across campus. There are many little places on campus where Dr. Beal planted the trees that are still there. So it's not just in terms of research, but actually in terms of our campus environment, the beauty of our campus, we can attribute to Dr. Beal. At any time in its history, MSU, during any one of its numerous financial crises, could have stopped funding the herbarium or the botanical garden. That's certainly valuable real estate in both of those institutions, but MSU, I think because of Dr. Beal's foresight, has had the wisdom to continue to invest in these bits of scientific infrastructure that Dr. Beal started or contributed.